video we are going to study about the development of female gametophyte. The female reproductive part of the flower is the pistil or carpel. The pistil or carpel can be single or it can be grouped one. If it is single one, it is called monocarpellary. Monocarpellary is the common examples as you find in case of mango, in case of wheat and all. And in case of SYNCARPOUS, that is where number of carpels are there which are fused together, as the common example you might have done in class 11th dissection of China rose or Dhatura, where more than one ovule or capillaries were carpels were there. The carpels, when two or more carpels are fused together, this condition is known as SYNCARPOUS. SYNCARPOUS may have more than one chambers, whereas the APOCARPOUS, each pistil is separated individual, that is the stigma, the style and the ovary, the stigma, style and ovary. This is the example of wheat and this is the example of Michelia, which is given in your book. Most of the monocotyledon plants, they are APOCARPOUS, whereas SYNCARPOUS may be present in dicot, but not all. So that's all. Now if we study the cross section of a ovary, we will find there are some locules, and in each locules one or more ovules are there. Ovules are the containing female gametophyte, and we have to study about that, how the development takes place. So when the ovary is there and when ovules are in their raw form, then the structure is somewhat like this. It has got a 2-inch cell. This is called Megaspore mother cell, which is surrounded by a number of 2-inch cells, that is called nucellus cells. The nucellus cells and the megaspore cells, nucellus cells are actually well protecting it. The megaspore mother cell will now undergo, because it is diploid and it has to produce haploid gametophyte, it undergoes some meiotic division. The first meiotic division, it results the formation of 2 megaspores, haploid in polyploidy, and then the second meiotic division also takes place soon after that, leading to formation of 4 megaspore, that is called megaspore tetrad. Now in most of the angiosperm, out of these 4 megaspore, 3 megaspore degenerates, and the rest of the development takes place from 1 megaspore, and that is what is known as monosporic development. So monosporic development means after the 4 megaspore which are formed, only 1 megaspore from the mature gametophyte, the other 3 degenerates. Okay, this is the left one is single megaspore. Megaspore then undergo mitotic division, forming 2 nuclei. Remember, it is only the nucleus formed, not the cytoplasm is not divided yet. Then it undergoes second meiotic division and resulting the formation of 4 nuclei. They all are haploid and no cytoplasmic division taking place. The third mitotic division is also taking place. So 3 mitotic division are taking one after other and resulting the 8 haploid nuclei. Cytoplasm is not yet divided. Then the rearrangement takes place, reorganization takes place in such a way that 3 of the first of all these nucleus will change into cell, and 3 of the cell move toward the antipodal side, 3 will come towards the micropylar end, and 2 will come at the center. The 3 which are toward the nuclear end or antipodal end, they are called antipodal cells. The 3 towards the micropylar end, the lower 2 are called synergid, and the one which reside top is called ovum, and the central 2 are called polar nuclei. Here the 8 cells can be seen clearly. Then on further development you will find that the 2 polar nuclei fuse together and the nuclei come together into one cell. So here it becomes 2 haploid nucleus and makes single cell. So finally, that when a female gametophyte is developed, it has got 7 cells but 8 nucleus. And the structure is somewhat like this. It is if I rub it out and I can label them. So this is, as you know, stigma, style, and this is the ovary part, solan part. These are antipodal cell, polar nuclei, cell and two synergies, SYNERGIDS. This opening is called micropyle, and these structures are known as integument, and this attachment is called funicle, FUNICL. So this is the total structure of one single ovule. This is one ovule in one ovary, but often you will find there are number of ovules in one ovary. Like in case of papaya, you see there are number of ovules find that each ovule develop into a seed. And otherwise it is an example of mango, in which there is only one ovule and that ovule change into one fruit, one ovule, and you will find this entire structure enclosed in the ovary. The ovary after fertilization change into fruit, whereas the ovule change into seed, and the egg change into embryo. The pollen nuclei will later on develop into endosperm, and endosperm will provide nourishment to the growing embryo, and these cells will degenerate. Integuments will form the seed coat. When it forms the seed, then the integuments will form the seed coat. There are two integuments, and accordingly there will be two seed coats, the outer and inner, and rest the stigma and style, they all will wither off or they will fall off. So this is what the structure is. Monosporic development you understood, your SYNCARPOUS, APOCARPOUS conditions you understood, the three parts you understood, and what is the fate of the different part of the pistil you understood. The next topic is pollination. Pollination, as you know, is an event where the pollen grains are transferred from anther to the stigma. As you know, like in animals, the plants cannot move. Secondly, even their gametes cannot move. Their gametes are also immortal. The plant itself is also immortal. Then they always require some external agent to transfer the gametophyte, that is the male gametophyte, that is the pollen grain, to the female gametophyte, that is the ovum. So how it is taking place, that is a very important thing. The pollination is of two types: self-pollination, that is within the same plant, or cross-pollination between the two different plants. Now self-pollination again is of two types, that is the AUTOGAMY and GEITANOGAMY. AUTOGAMY, that is when the pollen grains from the flower, that is the anther of one flower, falls onto the stigma of the same flower, then it is called AUTOGAMY. Number of plants are there which are known as the clistogamous flower. Clistogamous flower are those flowers in which the flower doesn't open at all. So there is no question that the outside agent can come in, and they will always show AUTOGAMY. Clistogamous is a dangerous situation because there is no case of external pollen reaching there, so it is almost there is no chance of new characters to evolve. The variation will be least available there, but it is also an advantage because it ensures that the pollination will definitely take place, because their organs, the anther and the stigma lies in the close proximity to each other. So in any case the pollen will definitely fall there and the seed will definitely form. So it is an advantage as well as it is a disadvantage. The other type of flowers are called chesmogamous flowers. These flowers are open flowers. They may have self-pollination or they may have cross-pollination. That is a different issue. GEITANOGAMY is the second case in which the self-pollination is taking place, but not within the same flower, but in between the two different flowers of the same plant. A flower is lying here, the second flower is lying somewhere down in the same plant. Pollen from here is transferred down, then it is called GEITANOGAMY. And GEITANOGAMY will definitely require some external agent, maybe a biotic or abiotic. And the third one is cross-pollination. Cross-pollination, in which the pollen grains are transferred from one plant to another plant, the flower of one plant to flower of another plant, but of same species. Though that is called cross-pollination. So that is all about the pollination. Now, agents of pollination, as I already said, that the plants themselves cannot move or they cannot take their pollen grains, so they are always dependent on some external agent. The external agent can be abiotic or biotic. Abiotic can be of two types, that is the wind or water. We shall discuss about the abiotic pollination first. The wind pollination and water pollination. In the previous class, that is in class 11th, we have studied about pollination in case of gymnosperm. That was essentially a wind pollination. All the gymnosperms were showing only wind pollination, whereas in the angiosperm you will find only a few species which are showing wind pollination, mainly grains, graminea family, maybe wheat, rice, and maize and all. So wind pollination, the pollen grain should be small, they should be produced in millions of number, they should be light, feathery, and the stigma should be sticky, and the flowers need not to be having any color or any sort of smell or any sort of honey or nectar or any other lurative agents. So these are all about the structure of the wind pollinating plant. You might have seen wheat, you might have seen maize. Just now I draw. So maize and wheat. The wheat, maize, you might have seen that the tassels which are hanging out, they all are stigma, and in these cases large number of flowers come together, so that the chances of receiving pollen grains increases. The wind pollinating, we already discussed, the flower need not to be colored, and in very often cases you will find that the male flowers are located higher in position and the female flowers are located lower in position, so the pollen grain can easily fly and reach up to the female flowers. The male and female, two different type of flowers will be there in the same plant or in the different plant rather. Abiotic pollination, second agent is pollination by water. Pollination by water is essentially shown by the aquatic plant, that is very much sure, but not all aquatic plants shows even pollination by water. Large number of aquatic plants are even showing pollination by other agents, maybe by insects or by birds or etc. Only those plants which are found, their flowers are found either on the surface or inside, they will show the pollination by water. There are only 30 genera in the, and mostly monocotyledon, which are showing pollination by water, or hydrophily it is commonly known as. So hydrophily is common only in 30 genera of monocotyledon plants, and for that the male gametophyte, that is the pollen grain, will be released, and that will passively move with the water current and reach to the female gametophyte. So the chances of their pollination decreases quite a low proportion of pollination take place, and that is why the wind pollination, water pollination is not a very common thing. The next is biotic. The biotic is taking place by insect known as entomophily. When the birds are involved, it is called ornithophily. When animals are involved, it is called zoophily, and when bats are involved, it is called chiropterophily. These are the agents. Insects are the most common. You might see honeybee, you might see other type of insect. There are large number of the bumblebee and other. There are large number of insects are there. During the season of mustard, you might have seen the small insects which often enter in our eyes. They all are actually meant for pollination, and pollination is greatly carried out by them. In chapter in the chapter of 8th chapter, we will study about the beekeeping. So they are playing a very significant role for pollination. So in addition, they give us honey. So that is another advantage. So pollination by insect is very common. Then pollination by birds are also seen in number of, like you might have seen hummingbee putting their beak into some of the plants, some of the flowers which are often bell-shaped flower. They are actually carrying out pollination. Invariably, they are not knowing that they are doing this pollination and all. Animals are also involved here. Zoophilia, like squirrels are there, moles are there. They are, and what is called the calotis, that is garden lizard is there. So they are actually carrying out this. Even monkeys are involved there. Even elephants are doing the pollination in some of the flowers. And bats in the night blooming flowers, which are white in color, and they are having these flowers. Naturally, whenever they have to involve some of their biotic agents, they have to provide some lubricative agents, that may have different colors, that may have different shape, and they should also provide some award in the form of nectar
or in the form of some insect which are living there because of those nectars and all. So these agents will reach there to the flower in order to get their rewards, maybe pollen grain they eat, maybe insect they eat, or maybe to collect the nectar and all. And when they go there, invariably their pollen grain stick into their body, and when the same insect go into some other flower, they transfer the pollen grain there. So this is how they are carrying out pollination. So what is more important is the summary of that is what are the structure or modification found in the wind pollinating plants. I already taught you that is the wind pollinating plants are not having color, not having any beautiful shape, they do not have any smell, they do not even bear any nectar, and the pollen grains are feathery, and large number of stigma come together in order to boost pollination. Male flower are generally found high in position and the female flower are generally found in the lower position. So these are all together in the pollination. Next video will study about the development of embryo and other things. Thank you. If you like the video, please press the like button and share it with the others, and if you are new on my channel, you have to subscribe it and press the bell icon so that you get whenever there is new video. Thank you.